Hello students, in this video I will continue the chapter molecular basis of inheritance. So now we are moving to the last topic in this chapter, that is DNA fingerprinting. But before that, let me explain some of the very important points related to the DNA fingerprinting, in that repetitive DNA or the satellite DNA. So what is this repetitive DNA? A small stretch of DNA which is repeated for several times is known as repetitive DNA. So this is a small stretch of DNA. A small stretch of DNA means a small sequence of nucleotides of DNA is repeated for several times, repeated for many times. So it varies from chromosome to chromosome. So this kind of repeated sequences, a small stretch of DNA which is repeated for several times, is known as repetitive DNA. Depending on the base composition, that is AT complementary base pairing as well as GC combinations. So based on the base pairing or the base composition as well as length of the segment. Length of the segment means based on the number of nucleotides found in that particular segment, as well as the number of repetitive units based on the number of repetitive units. That means the sequence which is repeated for how many times. So this is the number of repetitive units, and the satellite DNA is classified into many categories such as microsatellites, mini satellites, as well as macro satellites, etc. So this is regarding the important feature of the repetitive DNA. Now, these sequences normally do not code for any proteins, but they form a large portion of human genome. So this is a very important feature. See, this repetitive DNA do not code for any proteins. It will not code for any proteins. So this is the non-coding sequence. So this is a non-coding sequence. So already we discussed about the Human Genome Project. In that final we came to know that the entire sequence of nucleotides or the entire DNA molecule does not code for protein. Less than 2% of the total human genome codes for protein. So major portion of the human genome is composed of this repetitive DNA. So these repetitive DNA or the satellite DNA do not code for any proteins, but they form a large portion. They form a large portion of the human genome. So these sequences show high degree of polymorphism and form the basis of DNA fingerprinting. So the repetitive sequence or the satellite DNA shows high degree of polymorphism and it forms the basis of DNA fingerprinting. So the principle behind the DNA fingerprinting is the high degree of polymorphism of repetitive DNA. Then what is meant by polymorphism? If an inheritable mutation is observed in a population at high frequency, then it is known as DNA polymorphism. So normally from generation to generation, the genetic material undergoes mutation. That is a natural phenomenon. If an inheritable mutation is observed in a population, that too at high frequency, then it is referred to as DNA polymorphism. That means all the mutations may not express in the immediate generation. If it is observed in a population, that too at high frequency, then it is referred to as DNA polymorphism. Since DNA from every tissue such as blood tissue or hair follicle or the skin, bones, saliva, sperm, etc. from an individual show same degree of polymorphism. So this is another important feature. The same degree of polymorphism, they become very useful identification tool for forensic science. That means the specific chromosome in all the cells of an individual's body shows same degree of polymorphism. You know, all the body cells consist same number of chromosomes. A specific chromosome in all the body cells, body cells shows same degree of polymorphism. So that means we can isolate any cell from the body to isolate the DNA, to isolate the DNA. Further, as the polymorphisms are inheritable, so the polymorphisms are inheritable from one generation to another, that is from the parents to the children. DNA fingerprinting is the basis of paternity testing. It is the basis of paternity testing in case of disputes. So this is regarding the repetitive DNA or the satellite DNA. Now, let us study about the DNA fingerprinting. So the DNA fingerprinting technique was initially developed by Alex Jeffrey. So Alex Jeffrey started this technology, that is the DNA fingerprinting technology. So DNA fingerprinting is nothing but it is a technique of identification of an individual under molecular level. Under molecular level means DNA level. So a specific sequence of DNA is used for the identification of an individual. He used a satellite DNA as a probe that shows a very high degree of polymorphism. Just now I told about the polymorphism. A specific satellite DNA used as a probe that shows very high degree of polymorphism. And it was called as a variable number of tandem repeats or VNTR. So this VNTR, variable number of tandem repeats, is used in DNA fingerprinting. The technique as used earlier involved the southern blot hybridization. So this is a very important step involved in DNA fingerprinting, that is a southern blot hybridization. Southern blotting is nothing but it is a method or technique used for the separation of DNA fragments and using radio labeled VNTR probe. So this is another important feature. So in DNA fingerprinting, radio labeled VNTR probes are used for the hybridization process. So this is just an introduction to DNA fingerprinting. Now let me explain the important features of VNTR, that is a variable number of tandem repeats. What is VNTR? So it belongs to a class of satellite DNA referred to as mini satellites. So it is an example for mini satellite. As I told earlier, the mini satellite, micro satellite, macro satellite, this classification is based on the number of nucleotides found in that particular sequence, in that particular repeated sequence. A small DNA sequence is arranged randomly or tandemly in many copy numbers. So in case of VNTR, a small DNA sequence is arranged tandemly, tandem sequence or the tandem repeats in many copy numbers. The copy number varies from chromosome to chromosome in an individual. Just observe this statement again. The copy number varies from chromosome to chromosome in an individual. That means if a cell is found, this cell consists many number of chromosomes. It consists many number of chromosomes. So these are the metaphase chromosome. See, these are the chromosomes found in a single cell. The same way another cell is there. This cell also consists the same type of chromosome. There is no difference in the chromosome found in these two cells. But if you consider the VNTR sequence of this chromosome, that is totally different from the VNTR sequence found in this chromosome, that is the second chromosome. But if you compare this chromosome with the chromosome found in another cell, that is the same chromosome, here you can see the similarity. So the VNTR sequences found in this first chromosome is exactly similar to the VNTR sequence found in this first chromosome. And the VNTR sequence found in this second chromosome is exactly similar to the VNTR sequences found in this second chromosome, because both these cells belongs to the same individual. So within the cell, the chromosomes consist of different VNTR sequences, but the specific chromosome found in all the cells consists the same VNTR sequence. So the copy number varies from chromosome to chromosome in an individual within the cell. The numbers of repeat show very high degree of polymorphism. Already I told about this. It varies and it shows a high degree of polymorphism. The size of VNTR varies in size from 0.1 kilobases to 20 kilobases. So VNTR sequences are not same in all the chromosomes within the cell, so that it varies from, the size varies from 0.1 kilobases to 20 kilobases. After hybridization with VNTR probe, the autoradiogram gives many bands of different sizes. See, hybridization is by using radio labeled probes here, that I will explain while describing the steps involved in DNA fingerprinting technology. And after hybridization, we will have to take the X-ray photograph, that is called as autoradiography or autoradiogram. And in the autoradiogram, it gives the bands of DNA molecules of different sizes. Now, these bands give a characteristic pattern for an individual DNA. The bands give a characteristic pattern for an individual DNA. It is unique to the individual. So this is known as DNA fingerprint. It differs from individual to individual in a population, except in the case of monozygotic or identical twins. That means the identical twins are derived from the same zygote. So their genetic material is exactly same, 100% similar. So that's why the DNA fingerprinting is not applicable to the identical twins. The sensitivity of the technique has been increased by the use of polymerase chain reaction. So in DNA fingerprinting technology, we are using the assistance of or taking the assistance of polymerase chain reaction, because if we have very small number of DNA, if we have the DNA isolated from the single cell, then such DNA molecules can be amplified by using polymerase chain reaction. So the DNA from single cell is enough to perform the DNA fingerprinting analysis. For amplification, we are using the PCR. In addition to application in forensic science, it has much wider applications, such as in determining the population and genetic diversity. So this is regarding the important features of VNTR. Now, let us study the important steps involved in DNA fingerprinting technology. You should remember, there is no explanation for these steps in our textbook. The explanation is not given, so you can expect only the steps involved in DNA fingerprinting technology. They will ask just to mention the steps involved in DNA fingerprinting technology. So the first step in DNA fingerprinting technology, that is isolation of DNA. So isolation of DNA from which cells? See, for this purpose, we need to isolate the DNA from two samples. The isolation of DNA from two samples. So two type of samples are required. So which are the two type of sample? One is called as victim sample. Another one is a suspect sample. So in case of a crime, the sample collected from the crime spot is called as victim sample. That is called as victim sample. And the sample collected from the suspect. Suspect means criminals. Suspect criminals. Sample 1, sample 2. We can collect the sample from many number of individuals depending on the suspect. So the isolation of DNA from the victim sample as well as suspect sample is the first step. See, we can use the different type of cells. It may be in case of rape cases, the semen can be considered as a sample. The cells isolated, the sperms isolated from the semen can be considered as a sample, or even the tissues such as the skin, the hair with root. So different type of cells can be collected from the victim sample or from the crime spot. And even from the individuals, the blood sample can be collected, or other tissues can be, skin sample can be collected as a sample. So this is the first step in DNA fingerprinting technology. The second step in DNA fingerprinting, that is digestion of DNA by restriction endonuclease. So what is this digestion? Digestion is nothing but fragmentation. So this is nothing but the fragmentation. For this purpose, we are using the restriction endonuclease enzyme. See, I told we don't want the entire DNA molecule from the cell. We need a specific sequence from the DNA, that is called as VNTR sequence. So this VNTR sequence should be removed from the entire DNA molecule. For that purpose, we are digesting the DNA molecule. Digestion is nothing but fragmentation. And the fragmentation is achieved. The fragmentation is achieved by the help of restriction endonuclease enzyme. So only the VNTR sequences are required. So that's why the VNTR sequences are cleaved or removed from the entire DNA molecule by the help of restriction endonuclease enzymes. But see, this leads to the formation of a mixture of DNA fragments. So from that mixture of DNA fragments, we need to separate and isolate the VNTR sequences. We need to separate and isolate the VNTR sequences, and for that purpose we are using the technology that is called as gel electrophoresis. So gel electrophoresis is used for the separation and isolation of VNTR sequences from the mixture of DNA fragments. Now, in gel electrophoresis, all the DNA fragments will be separated, and we are focusing on the VNTR sequences. And these VNTR sequences are arranged in the gel electrophoresis according to their size. I told the VNTR sequences are not similar in length in all the chromosomes. It varies from chromosome to chromosome. So based on their size, the VNTR sequences will arrange in the gel slab in agarose gel electrophoresis. And all the DNA fragments are double stranded. So the next step in DNA fingerprinting, there is given in our textbook, it is given as blotting, but actually one more step is there, that is the alkaline treatment, during which the double stranded DNA fragments found in the agarose gel slab are treated with alkaline solution to convert the double stranded DNA fragments into single stranded DNA fragments. Just remember, the separated DNA fragments found in the agarose gel slab are actually VNTR sequences. VNTR sequences of victim samples as well as suspect samples. Only the VNTR sequences will be there. Now, all the double-stranded VNTR sequences of victim sample as well as suspect samples are converted from double-stranded to single-stranded by the alkaline treatment. Now, so single-stranded DNA fragments of both the victim sample as well as suspect samples are then transferred to a nitrocellulose sheet or a nylon sheet. The process of transfer of single-stranded DNA fragments from agarose gel slab to nitrocellulose sheet is called as blotting, specifically known as southern blotting. It is specifically known as southern blotting. So, in this step, transferring of separated DNA fragments to the synthetic membrane such as nitrocellulose or nylon sheet. So, this is an overnight process. Now, all the nitrocellulose sheet, this nitrocellulose sheet will have the single-stranded DNA fragments. So, these are the single-stranded DNA, separated single-stranded DNA fragments of a victim sample as well as suspect sample. So if you consider this as a victim sample, this is a suspect 1, this is suspect 2, like this. All the single-stranded DNA fragments, VNTR fragments, are found in the nitrocellulose sheet. So this is the fourth step given in our textbook. The next step is hybridization. As I told earlier, this nitrocellulose sheet consists of single-stranded DNA fragments. And these DNA fragments are actually the VNTR sequences of victim samples, suspect samples. These single-stranded DNA fragments are then hybridized by the process called as hybridization using labeled VNTR probe, labeled in the sense radio labeled DNA probes. So these radio labeled DNA probes are used for hybridization. So these radio labeled DNA probes are actually produced complementarily. These are produced complementarily to the VNTR sequences of victim sample. So the VNTR probes, radio labeled VNTR probes used for hybridization in this step are actually produced complementarily to the VNTRs of victim sample, not the suspect sample. The last step in DNA fingerprinting technology, that is detection of hybridized DNA fragments by using autoradiography. As I mentioned earlier, the radio-labeled probes used for hybridization are produced complementarily to the VNTR fragments of victim sample. The same radio-labeled VNTR probes are used even for the hybridization of suspect samples. Now, when the X-ray photograph of nitrocellulose sheet is taken, the regions of the presence of hybridized DNA will appear in the form of dark bands. So the dark bands found in the X-ray photograph or autoradiograph shows or it indicates the presence of hybridized DNA. Definitely all the VNTR fragments found in the victim sample or isolated from the victim sample shows hybridization, and they will appear in the form of dark bands in X-ray photograph or in the autoradiograph, because all the separated DNA fragments or VNTR fragments of victim sample undergoes hybridization, because we have used the complementary strands which are produced complementarily to the victim sample. The same radio-labeled probes are used for the hybridization of suspect sample. So the chances for the presence of hybridization is only if there is a complementary base pairing between the suspect VNTR strands as well as the radio-labeled probes used for hybridization. If there is a hybridization, such hybridized DNA fragments will appear in the form of dark bands. If the similarity is found between the autoradiograph of victim sample and the suspect sample, such suspected individual will be considered as the real criminal. So this is regarding the steps involved in DNA fingerprinting technology. Now, one diagrammatic representation is given in our textbook. Just observe this diagrammatic representation. See here, these are the different DNA chromosomes of the cells isolated from the crime spot. So these are the chromosomes of an individual who is actually involved in the crime, if it is a criminal case. So this is a sample isolated from the crime spot. This is chromosomes isolated from the cells got from the crime spot. And after the separation of VNTR sequences, just observe the chromosomes here. See, this is these two chromosomes represent the chromosome number 7. You know, the individuals are diploid, so two chromosomes will be there, paternal chromosome, maternal chromosome, and each chromosome consists a specific length of VNTR sequence. See, here it consists 11 nucleotides, here it consists 9, and it varies from chromosome to chromosome. So these two chromosomes represent the chromosome number 2. These two chromosomes, this pair represent the chromosome number 16. So like this, different chromosome consists different VNTR sequences. And these, after the separation of these VNTR sequences, they are processed, hybridized, and after hybridization, the X-ray photograph is taken. So this is the DNA fingerprint of the victim sample. And here you can see the presence of different bands. So these are the bands of DNA fragments. Hybridized DNA will appear in the form of dark bands. And here you can see two more bands. Now, so this is the sample isolated from, or the DNA isolated from the suspects, that is individual A. This is the DNA isolated from the individual B, suspect individual B. The same chromosomes are considered here, chromosome number 7, here also chromosome number 7. Even in the victim sample, we are using the same chromosome number, same chromosome, but the length of VNTR sequence varies. You can see here, so there is a variation in the length of VNTR sequence in this chromosome as well as in this chromosome. So it varies from chromosome to chromosome, not similar. So this is the separate sample, victim sample. So this is a suspect 1, this is a suspect 2. It varies. Now, after the isolation of these chromosomes or this DNA, their VNTRs are removed. They are converted into single stranded, then transferred to the nitrocellulose sheet, followed by hybridization, then X-ray photograph. When we took the X-ray photograph, when the autoradiography is done, so this is the autoradiograph of sample suspect 1 or individual A, and this is the autoradiograph or the DNA fingerprint of individual B. Now, after preparing the X-ray photograph or after preparing the DNA fingerprint, that DNA fingerprint should be compared. That DNA fingerprint should be compared. So just see here. This is the DNA fingerprint of a victim sample. This is a suspect 1 or individual A, and this is a DNA fingerprint of a suspect 2 or individual B. Just find the similarity between the samples, victim sample as well as suspect samples. If the similarities are found, such individual will be considered as criminal in case of criminal case. So just compare C and A or victim sample and suspect 1 sample. The first band is not same. Here there is no dark band, so this is wrong. So here also there is no similarity. So these two bands are similar. So all the other bands shows dissimilarity. So definitely we can come to conclusion that A is not the criminal. Now compare C with B or victim sample, the DNA fingerprint of victim sample with the DNA fingerprint of suspect 2. So first band exactly same, so there is a similarity. Even the second band is also same, there is a similarity. Third one is also same, fourth one is also same, as well as fifth one also same, even the sixth band also same. So by this we can come to conclusion that DNA isolated from the individual B is showing the similarity with the DNA isolated from the sample isolated from the crime spot. It means that individual B is the real criminal if it is a criminal case, because this sample, the victim sample, is actually belongs to the suspect 2 individual or individual B. So this is regarding the steps involved in DNA fingerprinting technology. So this is about the last topic in the chapter molecular basis of inheritance. So till now we discussed all the important topics mentioned in this particular chapter. As I mentioned at the beginning itself, this is a very important chapter for your theory examination as well as for the competitive examination. By this I will end this chapter. Thank you.